Good morning, everyone. Thank you for joining us at our webinar, Unlocking Food Authenticity, Molecular Biology Techniques and Bioinformatics Insights. I'm Cristina Ferretti, and I'll be your host in today's session. I'm the Division Manager of the Molecular Biology and Microbiology departments. I have a degree in pharmaceutical chemistry and technology, and I've been working in Neutron since 2000. I will give you a brief introduction on food authenticity, then our speakers will delve into the topics of the day. These are our speakers. Desi Burato is a molecular biology R&D specialist. She has a degree in biotechnology, and she has been working in Neutron since 2019. Loredana Longo is a molecular biology R&D specialist. She has a degree in biology and a PhD in biotechnology and molecular medicine, and she has been working in Neutron since 2007. Leila El Boulami is a bioinformatician. She has a degree in bioinformatics and joined Neutron about 1 year ago. This is our agenda. We'll start with a brief presentation of our corporate and of Neutron. After a brief introduction on food authenticity, we will delve into the main molecular biology techniques and the use of bioinformatics to detect food fraud. At the end of our webinar, we'll have a Q&A session, so feel free to use the chat box or the Q&A section to ask questions. Let's start with a brief introduction about our group. Cotecna is a Swiss-based company headquartered in Geneva with offices and labs across the globe. It is a leading provider of testing, inspection, and certification services. Today, there are more than 7,400 employees with more than 180 offices and laboratories. Our trusted network of professional and certified laboratories provides expertise across our 5 key sectors, namely agriculture, food safety, metals and minerals, consumer products and system certification, and ESG solutions. Neutron is the biggest laboratory of Cotecna Group, located in Modena, in the north of Italy. More than 550 employees work in 10,000 square meters of laboratory, managing more than 1,000 samples per day, with more than 5,000 accredited tests. These are the fields of testing activity which Neutron has activated during its 50 years of history in relation to the market and in relation to the customer requests. Now let's start with the definition of authenticity. According to this definition, a product is authentic when its identity, composition, and origin match the claims made on its label. Unfortunately, food fraud continues to rise globally. It impacts both consumer trust and public health. Fraudulent practices such as substitution, mislabeling, dilution, counterfeiting, concealment, and unapproved enhancement are more and more common. Studies suggest that 10 to 15% of food products worldwide are affected by fraud, costing billions annually. This is a list of the most frauded commodities in 2024, and as stated by the organizations that contributed to this survey, botanical and animal origin fraud were the most reported type of food fraud in 2024, followed by the use of non-food substances and dilution. There are several ways to guarantee authenticity. First, through certifications like ISO and organic labeling, for example. Second, with the traceability systems, which track the product from the field to the final shelf. And most importantly, through analytical methods, which provide scientific evidence that the product is genuine. One of the main challenges in detecting food frauds is the complexity of food matrices. From raw to processed foods with multiple ingredients, identifying fraud can be difficult. There are many methods available, but traditional methods often fail when dealing with complex products, making it crucial to use more advanced analytical approaches. To effectively detect fraud, a multi-method approach and a statistical analysis are essential. Among the methods used to verify the authenticity, molecular biology methods are precise and reliable tools with high sensitivity and specificity. Before moving to Daisy Burato, I remember you to report your questions in the chat box or in the Q&A section. Please, Daisy, the floor is yours.
Good morning to everyone, and thank you, Christina, for your introduction. The first section of this webinar will cover target molecular techniques and their use for testing the authenticity of food products. Molecular biology techniques are fundamental tools for the study and analysis of DNA and proteins. They can be divided by category in protein-based or DNA-based methods. The protein-based methods are LC-MS, MS, and ELISA. They detect the presence of proteins and are therefore suitable for the analysis of any matrix that contains not degraded proteins. The DNA-based methods are PCR in all its variants, Sanger sequencing analysis, and its step forward, next-generation sequencing. All these methods rely on the detection of DNA sequences, so they can be applied to any matrix that contains enough and not fragmented DNA. They are used in food authenticity thanks to their high specificity and sensitivity. Furthermore, DNA remains stable under a wide range of food processing methods. In food testing, two analytical approaches can be taken, target and untarget. It is important to know the differences between them to be able to choose the most suitable testing strategy. Target techniques are hypothesis-driven, so we know what we are looking for. They answer the question, is ingredient X present in the sample? Untarget techniques are exploratory. We don't have prior knowledge of what we are searching. They ask the question, what is in the sample? Target methods are sensitive and specific. They are ideal when we are looking to confirm the presence or absence of a specific substance. On the other hand, they are not able to detect unknown substances. Untarget techniques are characterized by broader detection capability. And for this reason, they are used when the aim is to analyze the entire sample, searching for unknown adulterants. Due to their screening power, untarget techniques collect huge amounts of data and so require advanced results analysis. Let's first focus on target methods. The ELISA, or enzyme-linked immunoadsorbent assay, is a target protein-based technique used to detect specific antigens. Its applications in food testing involve the detection of allergenic proteins, for example, milk, egg, and soy, and the detection of species-specific proteins. It can be useful to confirm the declared plant, meat, or fish species, or detect adulteration. as the detection of cheaper substitutes, like, for example, chicken in beef products. ELISA works through an antibody-antigen reaction. The output is a color change in the well proportional to the amount of target protein present in the sample. The techniques offer the following advantages. It is highly sensitive, it is a quantitative method, and the test is relatively fast and cost efficient. Regarding to limitations, ELISA test relies on intact protein epitopes. So it could be difficult to use this technique in heavily processed food in which proteins can be degraded. Moreover, there is a high risk of cross-reactivity with similar proteins. As previously said, PCR, polymerase chain reaction, is a target DNA-based method. PCR is used for species identification and for the detection of GMOs, allergens, and pathogens. PCR amplifies a specific DNA region using primers designed for a particular species, allergen, or genetic element. With the term amplification, we mean the creation of several thousand copies of the target DNA. Let's take a closer look at the variants of PCR commonly applied in food authenticity testing. The first type of PCR was the endpoint PCR, in which DNA amplicons are visualized after the amplification reaction, performing gel electrophoresis. Real-time PCR monitors DNA amplification in real time using fluorescent probes. Often it is used in a regulatory setting for allergens or GMOs. Digital PCR allows absolute quantification of the target DNA. It is more precise, but it has a higher cost. Among the advantages of this method, we can underline for sure its sensitivity. PCR can detect very low quantities of DNA, even in highly processed samples. These make it suitable even when the proteins are degraded due to processing. Primer design is crucial to ensure the specificity of the assay. It is important that they target only the species or allergen of interest. PCR is a rapid test. Results are available in a few hours. It is a cost-effective method. Once optimized, routine analysis is affordable. While PCR is a powerful and reliable technique, there are a few aspects to keep in mind for optimal results. First of all, sample quality matters. Indeed, the performance of PCR depends on DNA integrity. In some highly processed or heat-treated foods, DNA may be more fragmented, which can affect detection sensitivity. Food matrices are typically complex samples that may contain ingredients such as fats, polyphenols, or additives, which can affect the PCR reaction. Therefore, there is a need for protocol optimization. Careful sample preparation and optimized DNA extraction and purification protocols help overcome these effects. This table focuses on the main feature of the target techniques discussed so far. First, it is important to highlight the different targets of the methods: proteins for ELISA and DNA for PCR, as well their actual application to food process metrics, which is limited for ELISA and effective for PCR. Another issue to consider is the possibility of setting up a tailored assay. Indeed, authenticity tests are performed on a wide range of matrices, and it may be necessary to develop custom tests. The development of an ELISA assay is very challenging because of the antibody production, which is hard-working and expensive. On the other hand, setting up PCR is easier, faster, and more cost-effective. Let's look at a real-world case of meat adulteration that shook the food industry. In 2013, food authorities across Europe investigated the authenticity of processed beef products. The aim of the investigation was to verify the presence of other undeclared meat species in products labeled as containing only beef. The testing laboratories carried out ELISA and PCR analysis on frozen beef lasagna, burgers, and other ready-to-eat meals. The results reveal that products labeled as 100% beef contain undeclared horse meat, in some cases up to 100% replacement, indicating fraud rather than accidental contamination. PCR tests confirm the presence of equine DNA, and ELISA tests reveal high amounts of whole protein. Some samples also contain pork DNA, raising concerns for consumers with religious or dietary restrictions. So, target techniques like ELISA and PCR are suitable when the target of the analysis is known, the regulatory compliance requires detection of a specific marker, and when routine screening is needed. These techniques represent essential tools in our analytical toolbox for food authenticity.
Thank you very much, Daisy, for your presentation. Now let's move for the second topic with Loredana Longo.
Good morning, everyone. Thank you, Cristina, for this introduction. Thank you, audience, for joining us. As we saw in the previous section, targeted techniques are great when we know exactly what we are looking for. But what happens when we don't have a clear target or when we need to investigate a broad spectrum of potential fraud? In these cases, untargeted techniques like Sanger sequencing and next-generation sequencing are powerful tools. These techniques allow us to explore everything in a sample without knowing in advance which species are present, making them particularly useful in complex matrices where multiple species might be present. Let's start with Sanger sequencing, one of the most widely used molecular techniques for species identification. The method follows these main steps. The first is the amplification of a conserved genomic region present in multiple species. These DNA regions are present in all organisms but exhibit species-specific features. Then DNA is sequenced, and the resulting sequence is matched to reference databases to determine the species. Sanger sequencing has many applications. First, it's frequently used to identify plant and animal species by sequence-specific genetic markers like RBCL or cytochrome oxidase gene. In addition, it's used for genetic identification of bacteria and fungi by sequence regions like 16-RNA for bacteria and ITS for fungi to identify species. It's also used for confirming point mutation and verifying results from other sequence methods. Finally, it plays a key role in phylogenetic studies by comparing DNA sequences between organisms. However, there are some limitations to this method. One such limitation is that Sanger sequence is suitable only for samples that contain a single species. Here's a quick overview of how NGS works. First, DNA is amplified to increase the quantity of specific regions of interest. After the library preparation and the sequencing steps, the obtained data are computationally processed to assure quality and then mapped to a reference database. Here's an overview of NGS and its key applications. Next-generation sequencing is a powerful tool used in many fields. One such application is food fraud detection. In this case, NGS is used to detect food fraud by identifying undeclared or replaced species in food products. Another important use is species identification. NGS helps identify different species in the same sample, which is useful in fields like ecology and biodiversity studies. NGS is also used to study microorganisms, for example, in the food context. Therefore, NGS is a versatile tool used to improve accuracy in fields like food safety and environmental research. Let's now take a look at advantages of NGS in food authenticity. First of all, NGS provides high resolution insights into all DNA present, making it possible to identify a wide range of species and contaminants in one test. Another key benefit is that multiple targets can be analyzed in a single run, making it ideal for testing food products with complex ingredient lists. Lastly, NGS is an ideal method for broad screening, as it does not require any prior knowledge for this sample content. However, despite its numerous advantages, it's important to also consider the limitation associated with NGS. The first limitation is its cost and equipment. NGS requires specialized equipment and can be more expensive upfront. Another limitation is the complexity of data analysis. NGS generates massive datasets which require significant storage and processing capabilities. Lastly, another important aspect to consider is data interpretation, which require advanced bioinformatics skills to evaluate the results. Sanger sequence and the next generation sequencing differ in several key aspects. One of the main differences is the kind of sample that can be analyzed. Sanger sequencing is used for single ingredient samples, whereas NGS can handle more complex samples, analyze multiple ingredients at the same time. Secondly, Sanger sequence generates one single sequence per run, while NGS can produce many different sequences from different species at once, making it ideal for high-throughput analysis. Lastly, Sanger sequencing uses simpler, more accessible instruments. It's ideal for small-scale projects where only a few sequences need to be analyzed and processed directly by the instrument. In contrast, NGS generates complex, large datasets require advanced computational analysis. Let's now look at a practical example of how Sanger sequencing is used for species identification. In this case, a seafood importer requested confirmation fish species in a shipment of fillets labeled as European sea bass. Sanger sequencing was performed on a mitochondrial marker, cytochrome oxidase gene, for precise species identification. The result was that the majority of samples matched European sea bass as declared. However, 2 out of 10 fillets were identified as Nila tilapia, a lower value species. In this case, Sanger sequencing was chosen because the sample was clearly presumed to belong to a specific species, making it an ideal method for precise confirmation. Now, let's look at the case where NGS was used for spice authentication. A client requested verification of the presence of adulterants in a sample of ground oregano. NGS sequencing was performed for species identification. In this case, we were able to confirm the presence of oregano as expected, but also detected additional species, including olive leaves and sumac leaves, which are common adulterants often used to reduce cost and imitate the appearance of real oregano. When selecting a sequencing method, the choice between Sanger sequencing and next-generation sequencing depends on the type and complexity of the sample being analyzed. Both techniques offer specific advantages depending on whether the sample is simple or complex. Sanger sequencing is ideal for identifying DNA from a single species. Is appropriate for analyzing specific parts of a plant, pieces of meat, or fish where only one species is expected. It's commonly used to confirm the presence of a specific species in a sample. Next-generation sequencing is suitable for complex or mixed samples containing DNA from multiple species. NGS allows the detection of multiple species or genetic variations in a single analysis. Is recommended for the analysis of processed or ground products, for example, mixed meat or fish, or mixed plant material, where broad screening of the matrix is required. In summary, NGS provides an excellent starting point for broad, non-selective screening, especially when the sample contains many or unknown ingredients. It allows to identify all species present in a sample. Once this screening is complete, confirmation techniques such as PCR are used to verify specific DNA sequence, ensuring the accuracy of the results. By combining both methods, we get a complete and accurate food authenticity test.
Thank you very much, Loredana, for your presentation. And now let's move to the last topic of the day with Leila El Boulami. Please, Leila.
Good morning, everyone. Thank you for joining today's webinar, and a special thanks to Dr. Ferretti for the kind introduction. As highlighted in the previous section, NGS is an incredibly powerful technology. However, to unlock its full potential and extract meaningful insights, it needs to be supported by bioinformatics. For this reason, I'd like to start with a brief introduction to the role of bioinformatics. Bioinformatics is a multidisciplinary field that combines computer science, statistics, and biology to analyze and interpret biological data, especially large datasets generated from genomics, proteomics, and other omics fields. When it comes to untargeted methods like NGS, bioinformatics plays a crucial role. Without it, analyzing such massive and complex data would be incredibly time-consuming. Moreover, bioinformatics enables to extract biologically meaningful information from this data. Finally, it allows us to automatize and speed up analysis that would require weeks or months of manual effort through automated pipelines. It was precisely thanks to a bioinformatics pipeline that we were able to develop an analysis applied to food authenticity. Following this sequence of samples, as just described, we obtain a FASTQ file containing millions of DNA sequences. This file is extensive and complex. For this reason, the following step consists in applying a bioinformatics algorithm that allows to manipulate the data and to retrieve representative DNA sequences for each sample. The last step of the pipeline involves aligning our sequences against a reference database, namely NCBI, which is open source. The result we obtain is the identification of the species present in each food sample. Following this brief overview, let's examine each step of the bioinformatics analysis in detail. The first step, of course, is the data preprocessing and quality control of the raw sequencing data. The demultiplexing is performed to sort the sequences by sample-specific barcodes. Then, the denoising process is responsible for the removal of sequencing errors, thus recovering true biological sequences. Moreover, a filter is applied to set a minimum acceptable abundance of sequencing reads to assure the statistical significance of our results. Following the initial data preprocessing, we are still faced with millions of sequencing reads that need to be effectively managed before any meaningful analysis can take place. To achieve this, the sequencing reads are grouped together through a clustering process based on a 99% sequence identity threshold. This clustering algorithm helps to reduce redundancy by combining highly similar sequences into clusters. For each cluster, a representative sequence is then selected, which serves as a proxy for all the sequences within that group. This step is essential because it simplifies the dataset while preserving the key biological information, making downstream analysis more efficient and accurate. At this point of the workflow, data is cleaned and DNA sequences are ready to be aligned against a reference database. BLAST is the preferred algorithm for sequence comparison, thanks to its speed and accuracy in species identification. BLAST compares our representative sequences against NCBI, which is a comprehensive open source database, and as a result, we obtain the closest matches. Here is a practical example of a BLAST output. As shown, the algorithm computes sequence identity and coverage percentages, generates a score, and based on these metrics, enables species identification. Using open databases, such as NCBI, in food authenticity is key. First and foremost, transparency is a great advantage. Open databases are accessible to anyone, which means that any laboratory can verify the analysis independently. This builds trust and confidence in the results. Secondly, open databases support reproducibility, a fundamental principle of scientific research. Another important aspect is reliability. High-quality open databases are usually maintained by the scientific community, and they are regularly updated and curated. This ensures that the reference sequences used for the identification are accurate and reflect the most recent data available. On the other hand, proprietary or closed databases come with several limitations. Since access is restricted, it's often impossible to verify how the analysis was performed or what references were used. These systems may also rely on smaller, less diverse datasets, which can introduce hidden biases and compromise the accuracy of species identification. For all these reasons, choosing open and well-maintained databases is critical when the goal is to ensure scientific rigor and confidence in the results. It's important to underline that the results we obtain reflect the detection of the full range of organisms present in the food products. This means that even unexpected species that indicate food fraud or contamination are identified. Furthermore, the results are qualitative. Species abundance cannot be determined due to variable DNA amplification. Validating a bioinformatic pipeline is a crucial step to ensure that the results it produces are both accurate and meaningful. In our case, we performed internal validation by analyzing a series of known samples. The goal was to define an optimal read threshold that would allow for accurate identification while minimizing false positives and false negatives. We tested different thresholds. A low read threshold turned out to be too permissive. It introduced a significant amount of statistical noise and led to the detection of many false positives. On the other hand, a high read threshold was too stringent. It reduced noise but failed to identify some ingredients that were indeed present in the samples. Through this iterative process, we found that a threshold of an intermediate number of reads represented the optimal balance. It allowed us to correctly identify the ingredients present in the samples while maintaining statistical significance. Based on this internal validation, the pipeline using the selected read threshold was set as our final validated workflow, ensuring both sensitivity and specificity in our analysis. To demonstrate the strength of our bioinformatic pipeline compared to other commercial solutions, which rely on closed databases, we conducted a direct comparison using a mock mixture composed of known ingredients. This mixture included cumin, caraway, which is a known adulterant of cumin, sage, hazelnut, myrtle, and lentil. As you can see from this table, our pipeline successfully identified all the expected species. In contrast, the commercial pipeline correctly identified cumin, but only assigned sage and hazelnut at the genus level, without reaching species level resolution. Two species were not identified at all, likely because they were absent from the closed database. Additionally, lentil was reported with a slash code, indicating ambiguity in the identification. These results highlight a pivotal advantage of our approach. By using an open and well-curated database, we ensure broader taxonomic coverage and a greater accuracy. Moreover, every result is supported by expert interpretation, providing reliable and meaningful outputs for our clients. To conclude, let's explore the evolving role and future perspectives of bioinformatics in authenticity testing. Being a fast-evolving field, bioinformatics is expected to have an increasingly critical impact on ensuring food product integrity. The future perspectives include the expansion of bioinformatic tools beyond molecular biology. For example, they can be applied to other types of data, offering more comprehensive analysis approaches. Another key direction is the integration of artificial intelligence and machine learning. These technologies will enable faster and more in-depth interpretation of complex datasets, enhancing both prediction and accuracy. Finally, as analytical technologies continue to advance, bioinformatics will play an increasingly central role in supporting transparency, detecting fraud, and validating product claims, making it an essential tool in ensuring trust and reliability in the food sector.
Thank you, Leila, Daisy, and Loredana for your interesting and scientific presentations. Now we'll open the floor for a Q&A section. We have selected some of the questions that you sent us during the webinar registration. For the question received through the chat box, we will provide the answers via email. We'll start with the first one. Are there any official methods for the detection of food fraud? Daisy, could you please answer this one?
Yes, of course. Let me provide a bit more detail on that. There are currently only a few official methods specifically dedicated to detecting food fraud. For example, ISO standards exist for fish species identification at the international level, and CHEN standards cover mollusks within Europe. If anyone is interested, I'll be happy to share the references of these standards.
Thank you, Daisy. The second one is, what are the detection limits of NGS in the context of food fraud analysis? Loredana, since you previously discussed NGS, would you mind addressing this question?
Sure. Let me clarify that for you. So, detection sensitivity varies depending on the matrix and the composition of the mixture. Some matrices may allow for more efficient DNA amplification, while others can pose challenges due to the presence of inhibitors affecting DNA extraction or amplification. So, based on available studies and validation tests, the sensitivity of NGS for fraud application generally ranges between 1% and 5%, which is consistent with the intended purpose of this analytical approach.
Thank you, Loredana, for this insight. The third question is, NGS, is NGS a quantitative method? Can it determine the amount of each species present in a sample? Leila, since you mentioned this topic earlier, can you please share your thoughts on it?
Of course, that's a very common question we receive. Well, NGS is generally considered a qualitative method in this context. While it can identify the presence of different species in a sample, the number of sequencing reads doesn't reliably indicate the actual quantity of each ingredient. This is because, as Loredana has just said, DNA from different ingredients can amplify at different rates depending on the matrix. Therefore, it's not typically used as a quantitative tool for food fraud analysis.
Thank you, Leila. There's another question for you. Considering that you introduced the topic of the databases earlier, are there any reliable databases that are publicly accessible? Can you tell us a bit more?
Thank you for the question. There are indeed several publicly accessible databases, including NCBI, Unite, and BOLD. Among them, NCBI stands out as the most well-documented and complete, both in terms of the information it provides and also the wide range of organisms it covers. For this reason, we chose to use NCBI for our analysis.
Thank you, Leila. Moving to another question. Are there any methods other than molecular biology techniques that can be used to verify authenticity? Daisy, can you provide an answer here?
Yes, for sure. In addition to molecular biology techniques, Neutron offers chemical analysis methods to verify authenticity. These chemical tests help identify specific substances and provide an extra evidence to support the results from the molecular biology. So by combining both approaches, the chemical one and the molecular one, Neutron ensures a more complete and reliable verification process.
Okay, thank you very much, Daisy. The last question is, what are the most suitable methodologies for assessing fraud in spices and teas or infusions? Who would like to address this point?
I will take this one, Cristina. So, NGS is the most suitable methodology in this case. Since we are working with complex process matrices that contain multiple species at once. This type of analysis can properly applied to spices, teas, and herbal infusions, provided that the DNA can be extracted in enough amounts and with sufficient quality. So, the performance of NGS is highly depend on DNA integrity and purity, which may be affected by factors such as heavy processing, drying procedures, or the presence of compounds that inhibit amplification.
Thank you again, Loredana. We've received some questions in the chat box, so let's see one of them. For example, do you only perform product authenticity analysis, or can you certify a company product similar to SGF, Safe Global Fair? Please, Daisy.
We, as Neutron, perform authenticity analysis, but as Cotecna, we can certify our company.
There is another one that we can answer. While working in a Molbio lab, one concern is to receive the samples in correct condition for protein or DNA extraction. Do you advise your customers to send samples in some suitable condition, fresh or specific day old only, as this later affects TAT?
I can answer to this one.
Thank you.
So if the product is perishable, we absolutely recommend sending it under appropriate storage conditions, of course, because DNA can be degraded from an uncontrolled growth of bacteria flora.
Thank you very much, Leila. We are at the end of our webinar. We'd like to remind you that we will be responding to your questions via email in the coming days, or you can reach out to us at the following contact details. We hope you find this webinar helpful. A big thank to our speakers, Daisy, Loredana, and Leila, for sharing their expertise. Thank you to Dana Sanchez and all Cotecna team that helped us in the organization of this event. And last but not least, thank you to all of you for joining us. We will send out an email in the coming days containing a survey form, a recording of this webinar, and the relative presentations. Feel free to contact us for other questions and requests. You can find here Cotecna and Neutron contact. Have a great day, everyone. See you in our next webinar. Thank you. Bye-bye.